Life Sciences, Week 6, Video Lecture 12, Human Cells, Organelles. Okay, class, so now you've finished exam 3, so we're done with our microbiology, in which we've been largely looking at bacteria. Now we're going to kind of backtrack a little bit and look at cells. So we started looking at cells when we began our biology section, and we didn't, well, we saw a little bit of what was happening in plant cells. Mr. Anderson took us through a little thing on photosynthesis to see, some of what's happening in plant cells. So now we are switching our focus to animal cells, and that will be the focus for the rest of the quarter for us. So in this video, this is what we're doing. List and describe the 3 major parts of an animal cell, and give a brief description of the major cell organelles and the function of each. So here is a cell. This is really a lovely depiction of a cell that we have no cell that really looks like this, but this is, this looks really pretty. So recall, please, if you would, for me, that the definition of a cell, oops, I have a cat on my keyboard here. A cell is the basic structural and functional unit of living organisms. A cell is the smallest unit of a living organism that displays all characteristics of life, which means we should see movement in a cell, we should see growth in a cell, we should see reproduction in a cell, we should see responsiveness in a cell, etc. All of those things we talked about as far as the definition of life. So, getting into animal cells here, let's take a look at the 3 parts of a cell. So first of all, why am I making a distinction between animal cells and plant cells? Well, because they're different, okay? So that's a good reason to make a distinction. They're not very different. They're both eukaryotic cells. In animal cells, we're not going to see photosynthesis happening, so all of those aspects of the cell that are carrying out photosynthesis, we're not going to find that in an animal cell. Also in an animal cell, we're not going to find a cell wall the way that we do in plant cells. Plant cells build a cell wall around every individual cell, or most individual cells, and that's part of the structure of the plant. So animals don't do that. So we're looking at animal cells, and so why animal cells? Well, of course, because we are animals. So what we're really looking at is our own cells. And in this process of looking at cells, oftentimes we'll see images like this one of sort of a generic cell. And so, like, if we were looking at a muscle cell, it doesn't look anything remotely like this. If we looked at a brain cell, same thing, not remotely like this. In fact, I can't think of a cell in the body that looks like this at all. But the point is, what we look at a lot of times is sort of this generic version of a cell, so that we can see all of the parts of the cell. And then, when a cell is specialized and carrying out some specific function, it may have a lot more of a certain organelle, or more inclusions, or whatever, or it's going to be shaped in a specific way, that kind of thing. Anyway, so let's take a look at our 3 parts of a cell. So the first of the 3 parts is the plasma membrane, which is also called the cell membrane. And we've looked at the cell membrane before because we've looked at, well, sort of we have, because we learned about these molecules that make up the cell membrane. So we know that the cell membrane is made up of largely of phospholipids. Those are the ones with like the pink balls with the blue kind of squiggly things coming down from them. So what we see here is a phospholipid bilayer that makes up the majority of the plasma membrane or cell membrane. And why is it called a plasma membrane? Because it's called plasma membrane because it's made up of it's like this it's a unit that's made up of lots of individual parts, and those individual parts are constantly changing position and moving and being part of the cell membrane, and then not being part of the cell membrane because they go and be part of something else. So it's very fluid and dynamic. In fact, the model that we use to describe all of the movement and activity of the cell membrane is called the fluid mosaic model. So our cell membranes are a fluid mosaic. It's made of many different parts, like the proteins, the cholesterol, the phospholipids, but it's a fluid mosaic. So anyway, so that's the plasma membrane or the cell membrane. Its function is to regulate the flow of materials in and out of the cell. So the cell membrane is permeable to some things and not permeable to other things, and it changes. It has what's called selective permeability, so it changes what it's permeable to at different times based on the needs of the cell and based on what the rest of the body is communicating with it. So it does that. So basically that means it creates this clear boundary between what is the inside of the cell and what is the outside of the cell, and then it regulates what is on the inside and what is on the outside. And so that's one function. It also helps to maintain proper internal and external environment for normal cellular activity. So again, it's this boundary that designates the inside of the cell from the outside of the cell, but it also regulates the flow of materials in and out of the cell, so it has control over what materials are inside and what materials are outside. And so in that, it maintains the proper internal and the proper external environment for normal cellular activity. And then it also facilitates communication, both among cells and between cells. Between cells and their external environment. So when cells communicate with each other, it is through their cell membranes. So one of the features of cell membranes are receptors. And so when those receptors, depending on what those receptors receive, like if it's a hormone or which hormone it is, or if it's a neurotransmitter from the nervous system, any of those things are going to communicate something to the cell. And all of those things are going to communicate something different to the cell. So those are the functions of the cell membrane. We will do a whole lecture on the cell membrane next. I think that's our next lecture. So that's the first part of the cell. The second part of a cell is the cytoplasm. Now the cytoplasm actually has three parts to it. So the three parts of the cytoplasm are the cytosol, which is the fluid, the intracellular fluid, the organelles, which we're going to look at in more detail in this video, and the inclusions, which are temporary storage, things that are being temporarily stored in the cell. So inclusions, like if we were looking at plant cells, inclusions might be, well, plant cells almost always have inclusions of water, but also plant cells, like if it was a cell that makes up the petal, it might have inclusions of pigments that make the petal be the color that it is. And if it's, or whatever, inclusions that whatever, that's a good example. So, but other things that a plant might store, there could be inclusions of that. In animal cells, inclusions, the best example of inclusions would be like in liver cells, or one example anyway, in liver cells and in skeletal muscle cells, we store glycogen. And so glycogen is a molecule that's stored in those cells. And so that is an example of an inclusion. Also, like in adipose cells, our fat cells, the triglycerides that are stored in there are considered inclusions. Okay, organelles, we're going to take a look at those. Cytosol, that's the fluid. I think I said all this, right? Okay, so the cytoplasm is made up of those three things, the fluid, the cytosol, the organelles, which are the little organs of the cell, and the inclusions, the things that are being temporarily stored in the cell. That's the cytoplasm. The third part of an animal cell is the nucleus. So if we look at the same diagram, we see that large purple part that has a more concentrated purple circle inside of it. That's the nucleus. So the nucleus stores the DNA, and that's where DNA activity takes place. So if we're copying our DNA, or if we are making RNA from the DNA, that kind of activity happens inside the nucleus. So it would be kind of easy to think of the nucleus as another organelle, but it's actually considered its own thing, different from being an organelle. So another way that we can define cytoplasm is that cytoplasm includes everything between the cell membrane, or the plasma membrane, and the nuclear membrane. So the nucleus is defined by a membrane also, and so everything between those two membranes is the cytoplasm. All right, so let's go ahead and take a look at our organelles. The first organelle we're going to look at here is the cytoskeleton. So the cytoskeleton is exactly what it sounds like. So the word part cyto means cell, and skeleton means skeleton. So the cytoskeleton is the protein structure of the cell. We have 3 different size proteins that make up the cytoskeleton of cells. But literally, this is making the physical structure of a cell, or it's helping a cell to hold its shape and maintain its shape. And it supports the cell membrane and those sorts of things. And then if this is a cell where a lot of movement is happening, there will be aspects of the cytoskeleton that are supporting either carrying out, well, yes, both carrying out that movement, but also supporting that movement, like protecting the cell membrane against breakage in an area where there's a lot of movement happening. So the 3 parts of the cytoskeleton these are basically divided by size. So the smallest are called microfilaments. So we see those on the bottom of this diagram. It's the light green thing. So you can see that microfilaments. So what they're showing us an example of here in this particular diagram is actin. So actin molecules make up a microfilament of actin. So the actin molecules are the green balls, and then they're all strung together. The light green balls at the bottom, they're all strung together to make a fiber called a microfilament, and but each of those green balls is a complete actin molecule, and actin is a protein. So then we have this fiber of protein made up of all these individual proteins. So we could say, oh, look at that. That's the quaternary structure of that protein, because we have these complete proteins that are the actin, those green balls, and then we put them together in a specific way, and we make these fibers, and those are the microfilaments. So what do microfilaments do? They're the smallest of the 3 types of the cytoskeletal proteins, and they're located just inside the cell membrane. So they support the cell membrane. So if we looked at, let me go back to that diagram in the cell and see if we can see it here. Kind of. So do you see little like hair-like structures? These like fibers, these thin fibers all through here. Those would be the microfilaments. So they are just inside the cell membrane, and they're supporting and creating the shape of the cell membrane. And also, let's see if we can zoom in more here. These little structures that are sticking off of the cell membrane, these are called cilia, which is just a structural feature of some cell membranes. Each cilia is supported and created by a microfilament. So there's a microfilament inside each of those, not cilia, microvilli actually is what those are called. Those are called microvilli. And so inside every microvillus is a microfilament. So it's a protein structure that helps to create that particular shape of the cell membrane. So let's see. So they move and they shape the cell membrane, so those are the microfilaments. So let's go back to our other slide here. Okay, so we looked at microfilaments. The intermediate size type of fiber are called intermediate filaments. So of course intermediate means like middle, right? So those are the middle-sized filaments. So intermediate filaments, they're actually showing up on top on the right on this diagram. Intermediate filaments are thicker and stronger than microfilaments, so they're going to provide more support in areas of mechanical stress, and they stabilize the positions of some organelles. So, for instance, we'll see some organelles, but like the endoplasmic reticulum, for instance, needs to stay oriented to the nucleus, and so intermediate filaments are going to help with that orientation and the connection between those organelles that need to stay in close proximity to each other. So that kind of thing is what intermediate filaments are doing, but also creating more support. So like if there's a part of like say in a sperm cell where there's a flagella that's moving, that movement of the flagella, the cell membrane around there takes a lot of stress, and so there are going to be intermediate filaments around there rather than just the microfilaments to support and help hold the structure of the cell membrane. And then our third and largest type are called microtubules, and microtubules do all kinds of things actually. So they're going to be the ones that form much larger extensions of the cell membrane, like in a nerve cell, for instance. Nerve cells have dendrites and they have axons, which are big long extensions off the cell. And even back to the sperm cell example, the flagella of a sperm cell, inside there, we're going to have microtubules because it's a longer extension, and so it needs more support. And so same thing with a nerve cell. The axon off of a nerve cell is a very long extension off the cell, so it needs more support. So we're going to have microtubules inside of there. All right, so those are the cytoskeleton. So 3 different size protein fibers that support the shape and structure and movement of cells. That's our first organelle. Our next organelle are the centrosomes. Centrosomes actually are not found in every single cell, but they are found in any cell that is destined to divide. So not every cell is destined to divide again, because some cells, once they differentiate and take on their mature adult activity, this could be in a kid, of course, too, but the activity that cell is destined to carry out for the rest of its life, once it's doing that, and if it's not going to divide again, then it won't have centrosomes. But any cell that is going to divide again will have centrosomes. So, of course, like all of our stem cells are going to have centrosomes, because stem cells, what they do is they divide and give rise to those daughter cells that will differentiate into the mature cell of that tissue. So centrosomes are made up of centrioles, which we see on the left here. Centrioles are this sort of the cylindrical shaped organization of microtubules. So here we're seeing microtubules, and we just talked about them. And so there are 2 sets of these of this organization of these microtubules, and they hang out at right angles to each other. So there's one going one direction, and then the other one is kind of crossed at a right angle. And so those are the centrioles. But what a centrosome is those centrioles plus sort of a halo of enzymes around them. So in the diagram, in the right part of the diagram, you can see an arrow pointing to centrosomes. So you can see within each of those places where a centrosome is indicated, you can see the 2 centrioles, and then you can kind of see this halo around it. And so the centrosome is the whole thing, is that halo plus the centrioles. And that halo is basically made up of enzymes that are needed when the centrosomes need to function. So centrosomes function as cell division. So what that means is most of the time they're hanging out doing nothing at all. But when it's time to divide a cell, then the microtubules that are part of the centrioles extend, and they reach way out all the way to the center of the cell where the what are they called? The chromosomes are all lined up, and they grab a hold of the chromosomes and pull them to opposite ends of the cell. So essentially that divides the nuclear material, divides the DNA. So then we have equal copies of the DNA on each end of the cell. So that's just one part of cell division that, if we get to that point in this class, if we look at cell division, then we'll see that happening. But that's what centrosomes do, is they pull the DNA to opposite ends of the cell, so they're really only active for a brief time during cell division. Okay, our next organelle is the ribosome. The ribosome is pretty darn active all the time. So remember that ribosomes are the only organelle that we find in prokaryotic cells, and we find plenty of them in eukaryotic cells. So ribosomes make proteins. So what they do, so they're actually made up of 2 individual parts. So in this diagram we see this kind of large orangey glob on top, and then kind of a smaller white and light bluish kind of glob on the bottom. So the orangish glob on top is the large subunit of the ribosome, and the smallish glob on the bottom is the small subunit of the ribosome. So when the ribosome is not actively producing proteins, those 2 subunits are separate from each other. And then when it's time for that ribosome to produce a protein, then they get together, and they also get together with, you see that purple sort of sneaky looking thing across the middle of the diagram. or in between the two subunits, and it's labeled mRNA. That is messenger RNA, and that's the RNA molecule that contains the information on how to make the particular protein that we see this ribosome actively making right now. So the two subunits plus messenger RNA get together, and then it's the ribosome's job to read that RNA molecule and allow for what are called transfer RNAs, and we can see them, that's that green sort of T-shaped thing in the middle, and it's labeled tRNA. So that's a transfer RNA. Transfer RNAs bring in amino acids, and so so the ribosome reads the sequence of nucleotides on the messenger RNA, and the transfer RNA brings in the correct amino acid that's called for in the right sequence, and then the ribosome puts those amino acids together in order. So when we were looking at proteins and the structure of proteins, the first layer, I guess layer you could call it, of structure of a protein is the primary structure, which is just a sequence of amino acids. That is what the ribosome takes care of. It puts the amino acids in the right sequence. So that's the job of the ribosome. The next two organelles that we're looking at are the endoplasmic reticulum. There are two types of them, and one of them is closely associated with ribosomes. So what we're looking at here in this diagram is we've zoomed way into the cell. This the thing in the diagram, well, it's labeled, well, you see nuclear envelope labeled, we see nuclear pore labeled, so that's the nucleus. Now the nucleus is opened up, and there's actually no DNA inside there, I think, just for the simplicity of the diagram, but DNA would be inside of that. Okay, so that's the nucleus. So then attached to the nucleus we have a rough endoplasmic reticulum that we see this more toward the bottom of the diagram, and then a smooth endoplasmic reticulum that's closer to the top of the diagram. But you know what's super weird? I just noticed this, they have the line pointing to the wrong place. The smooth endoplasmic reticulum is, that line should be pointing straight down. It's that, those things that sort of look like tentacles coming up, that's the smooth ER. But instead they have it pointing to the nuclear membrane. So anyway, so that's a little wrong on here. But anyway, the rough endoplasmic reticulum is the one that's toward the bottom, and it's called the rough endoplasmic reticulum, or rough ER, because the surface appearance of it looks like it's rough. And so the thing that makes it appear rough are ribosomes. And so those are the little purple balls that we see there. Those are ribosomes that are embedded in the membrane of the rough ER. So we just looked at ribosomes. We know what they're doing. Ribosomes are assembling amino acids in the right sequence to make a protein. So they're doing that, the primary structure of a protein. So ribosomes that are embedded in the wall of a rough ER, they assemble proteins into that membrane space that you can see there, that sort of orangish membrane, intermembrane space. They put their proteins that they're making into that. And so then inside the rough ER, what happens is the proteins get their secondary and tertiary shape, and they get like packaged also. So, if a ribosome is making, 20 copies of the same protein, all 20 copies are going to end up in a same little packet in there, and they'll all be shaped and prepped and ready to move on to the next step, all inside the rough endoplasmic reticulum. So the rough ER has a lot to do with protein synthesis, but it's not the assembling of the proteins. That's what the ribosomes are doing. It's that next step. It's getting that protein ready to be active and making sure it's shaped correctly and all of that. That's the rough ER. The smooth ER deals with the synthesis of lipids. So whatever lipids this cell needs to produce, those are going to be produced inside the smooth ER. So there are enzymes in there that facilitate lipid synthesis. So if this was let's say this is a liver cell, and this liver cell needs to take some excess glucose and convert it to lipids for storage, that could happen inside the smooth ER. Or let's say this is just any old cell, and this cell is saying to itself, gee, I could use some more phospholipids for my membrane, then those phospholipids could be made in there. So whatever lipids this cell needs to make, those lipids will be assembled inside the smooth endoplasmic reticulum. Okay, so we have the rough ER and the smooth ER, and then one of the things that you'll see as we look at individual cells, although we're not going to do it in this class, is there are variations on the endoplasmic reticulum also in specific specialized cells. So like in muscle cells, we see something called the sarcoplasmic reticulum. So a sarcoplasmic reticulum is a variation on this endoplasmic reticulum theme, but it's something that stores a substance, a specific substance in a muscle cell. So endoplasmic reticular can be used for that as well, for storage, but they'd be specialized in special differentiated cells. Okay, what's next? The next one is the Golgi complex. So the Golgi complex is also called the Golgi apparatus, and what we're looking at here is the Golgi complex is the thing that's on the right. It's again a series of membranes, so it looks kind of like a smooth ER in a way. And it looks like it kind of has like bubbles coming off of it. So the Golgi complex, what it does is it does packaging and sorting in preparation for export from the cell. So if the cell is producing, say it's producing a fatty substance in the smooth endoplasmic reticulum, but that's something that's going to be exported out of the cell, then that substance will go from the smooth ER, and it will go inside a membrane that's formed from the membrane of the smooth ER to the Golgi complex. So then that membrane will merge with the Golgi complex membrane, and then the Golgi complex will say, what's this here? Oh, I see, this is a concentration of some particular, say it's a fat-soluble hormone. And so, okay, so I'll get this ready for export from the cell, and so it does that and packages it up, and then sends sends a vesicle pinched off from its own membrane full of that substance to the cell membrane, where it then merges with that vesicle merges with the cell membrane and spills the contents outside the cell. So that can happen with fatty substances. What we see in this diagram is it happening with proteins. So on the top diagram we see, or the top part, we see a transport vesicle that has pinched off of the Golgi complex. It's filled with it's just labeled soluble proteins. So that could be a hormone that is a water-soluble hormone, so those are made out of proteins. There are various things that it could be. It could be exosomes even. So this transport vesicle is filled up with many copies of the same protein. It's going to migrate to the cell membrane, merge with the cell membrane, and in doing so, it sort of like turns itself inside out and spits its contents outside the cell. So that's a secretion of that cell. And the bottom part, what we see here is the Golgi complex has prepared what's called a secretory vesicle. The top one, we're calling it a transport vesicle. And it's just a minor difference about what, it's not so much about what's contained in the cell, it's about, in that vesicle, it's about how the cell knows when to release that vesicle. So in a transport vesicle, the cell is just like, oh, this is just a substance that we're getting rid of, so I'm just going to send it to the cell membrane and get rid of it. Whereas a secretory vesicle isn't going to be released until the cell gets a signal to release it. So an example of that would be like if this was a nerve cell and it was producing neurotransmitters. So neurotransmitters are the chemical communication aspect of nervous system communication. So that cell, it's going to produce those neurotransmitters, put them in a secretory vesicle, and it will hover there near and so that's happening in the Golgi complex. And so the Golgi complex gets those packaged and ready to go to the cell membrane. And then they'll hover near the cell membrane in the secretory vesicle, and then when a signal comes from another cell, so it could be a nerve stimulation or it could be a hormonal signal, that will stimulate the release of the substance in the secretory vesicle. So at that point, that vesicle will merge, same way that the transport vesicle did, it will merge with the cell membrane and spill its contents outside of the cell. But the Golgi complex, we're talking all about these vesicles. The Golgi complex is the thing that makes those vesicles. So it packages whatever it is that's inside those vesicles. So it sorts things out and makes sure that each vesicle is filled with just copies of that same thing, and it labels it. so that the cell knows what it is and knows when and how and where to release it. So that's the Golgi complex. And what's our next one? Lysosomes. So lysosomes are little organelles found inside cells. We'll see a diagram here in just a bit where we see all of these, again in the whole cell, and they're labeled, so we'll see kind of the different size, because lysosomes are actually pretty small, but we just see this lysosome here by itself, so it's hard to tell how small it is. But what lysosomes are, or what they do, is they focus on intracellular breakdown reactions. So lysosomes are so that some word part always means body, lyso means cut. So this is a cutting body. In other words, this is a body that contains enzymes that break things down. So this organelle is made up of a phospholipid bilayer, like many organelles membranes are, because phospholipids are really good like that. They're good about separating water compartments. And so that's what we have. It's actually labeled plasma membrane here, which is the same thing as a cell membrane, but I don't know. Personally, I wouldn't call it a plasma membrane. I would just say that it's the membrane of the organelle. But it's not inaccurate to call it a plasma membrane. But it is made of a phospholipid bilayer. Inside this lysosome is a hydrolytic enzyme mixture. So we know what that means, right? It's enzymes, and it's enzymes that are doing their reactions through hydrolysis. So that's why it's called a hydrolytic enzyme mixture. So remember that hydrolysis is the cutting apart of molecules by inserting a water molecule between, into parts, in between two parts of the molecule to separate them from each other. So that's hydrolysis. And that's the kind of breakdown reactions that are happening inside lysosomes. So for instance, we see lots of lysosomes in white blood cells. In white blood cells, it's their job to take in a bacteria or some foreign substance that we don't want to have in the body, and take it in, put it into the lysosome, and break it down. So so a bacteria will be brought in by through the cell membrane in a specific way, kind of like what we were just looking at here, when we were looking at these transport vesicles taking things out. Bacteria can come into cells in the same way. So a white blood cell membrane is going to grab onto a bacteria that it wants to get rid of and bring it into the cell and form a vesicle around it like this, and then that vesicle will merge with the lysozyme, and so then the contents of that vesicle is then exposed to the contents of the lysozyme, and actually, the phospholipids that make up the transport vesicle merge with and then become part of the membrane of the lysozyme. So that's so you can see the sort of fluid mosaic thing that's happening, that these molecules that had been part of the whole cell membrane, now they're a transport vesicle, now they're part of the lysozyme membrane, or lysosome membrane, I mean. So anyway, so then once that bacteria is exposed to the hydrolytic enzyme mixture of the lysosome, then it gets broken down, so then it's essentially dead and no hazard or danger to the cell, and then its contents will get spit back out. And we might even use some of those things from the bacteria, we might use some of the molecules that made it up. That's the lysosome. Our next one is very similar looking. It's called a peroxisome, and it is essentially exactly like a lysosome except for what's contained in it. So it's the same idea, where it's a phospholipid bilayer that separates it from the rest of the cell. It's again a small organelle that contains this time an enzymatic mixture that, instead of doing hydrolysis, it does redox reactions. And we haven't talked about redox reactions yet, but redox reactions involve the moving around of electrons. And it's another way, essentially, to break something down. So what peroxisomes do is they're going to break down substances by taking hydrogen ions and connecting them with oxygen molecules to turn it into water molecules, or into hydrogen peroxide, which is where the name peroxisome comes from. Peroxisome means that it's a body with hydrogen peroxide in it. So it will form hydrogen peroxide, but then also safely break that down inside of it. So it does that to protect the cell from certain toxic substances, like free radicals are a great example of what peroxisomes are up to. So free radicals are can be highly destructive to cells, but are constantly made as a byproduct of ATP production. And so when those are accidentally yet constantly made from ATP production, we need to get rid of them, because otherwise they would be dangerous for the cell to have around, because they're very destructive. So the peroxisomes will take those on and break them down and make them not dangerous for the cell. So those are what the peroxisomes are doing. And I think this is our last one, the mitochondria. So we'll talk about mitochondria quite a bit here, actually, when we talk about ATP production. But that's what's happening inside of mitochondria, is ATP production. So both the Krebs cycle and the electron transport chain happen in the mitochondria. One happens in the matrix of the mitochondria, and one happens in the membrane, that inner membrane of the mitochondria. And so here's a little fun fact that's super cool about mitochondria. Mitochondria, it is thought that mitochondria are actually like their own living organisms. They are, because they're very much like prokaryotic cells. So they are like their own little organisms inside our cells. So you can kind of see they sort of look like prokaryotic cells. And so, but why would we say that? Why would we say that this organelle is actually like a prokaryotic cell? Well, one of the things about it is that it contains its own DNA. So see that DNA label at the bottom is pointing to two different rings there of DNA molecules. So so our own, whoops, what's going on? Our own DNA in the nucleus does not contain the information on how to make a mitochondria. Mitochondria contain their own DNA on how to make them. And they will reproduce themselves as necessary based on our ATP needs. And so, so it's kind of like they are, sometimes mitochondria are called the powerhouse of the cells, because they are the places where a whole lot of ATP production happens. So if we didn't have these mitochondria in our cells, we would not be able to utilize the fuel that we do for making ATP and oxygen the way that we do to make great numbers of ATP quickly. So we could still make ATP without mitochondria, but it'd be so slow. And our reality would look very, very differently, because we'd be making ATP so very slowly. But we also wouldn't need oxygen if we didn't have mitochondria. So I don't know, potentially that would be a plus, but maybe not. Okay, so mitochondria, that's where huge amounts of ATP production are happening, and we'll take a look at those guys a little more when we look at ATP production. So our last slide here is another diagram of the cell. So we can see all of these organelles that we just talked about. So up on top we can see a couple of mitochondria. We see some microfilaments there creating some structure inside the cell membrane. This cell is also shown as having microvilli, so we know that there are microfilaments inside that microvilli creating that structure. So we see a lysosome there. The blue ones are lysosomes, and we also see peroxisomes. So I'm going around kind of counterclockwise here. So lysosomes, where we have hydrolysis happening, peroxisomes, where we have redox reactions happening, but both of those are places where toxic substances are broken down. We see centrioles right there kind of in the center. So if we see centrioles, we know that this is a cell that is destined to reproduce itself. We don't really see that haze around it that makes the centrioles called a centrosome, but we see the centrioles, and they are part of the centrosome. We see microtubules, those larger protein structures inside the cell. We see a Golgi apparatus there. The red one is a Golgi apparatus. We see the smooth ER that's down a little bit from the Golgi apparatus. They're saying these are cilia on this cell, not microvilli, but cilia are simply larger than microvilli, but it's the same idea. It's a structure of the cell membrane. Let's see, doesn't this look like sort of an underwater world in here? Like you could go scuba diving and see all these things down underneath the water. Okay, smooth ER, so that's where lipid production is happening. We see the rough ER, we see ribosomes within the membrane of the rough ER, so we have protein synthesis and protein shaping and forming happening there. The next one is chromatin, which is just the form that DNA is in when it's not in its chromosomal form. So most of the time when it's hanging out inside the nucleus, it's in its chromatin form. And then the nuclear envelope and the nucleolus, which we haven't really talked about too much. The plasma membrane, which that's the cell membrane of the cell, and yeah, the nuclear pores, nucleus, that's all nucleus stuff. And then this is showing more rough endoplasmic reticulum on the other side of the nucleus. So those are our organelles. So we listed the three major parts of the animal cell, so the plasma membrane, the cytoplasm, and the nucleus, and then we gave a brief description of the major cells, of the major organelles of the cell. So yay for us. Your homework is, there is a coloring book page on Populi that is basically a diagram of a sort of a generic cell, and all you need to do is color it. So you'll color all the lysosomes the same color, and all the mitochondria the same color, and that kind of thing. So that'll just take you through looking at all of these organelles again. There are 2 pages to it. You just need to color the coloring page, but the other page has the description of all of the organelles. So that's there for your reference. But when you turn it in, you can just take a picture of the page that you colored and then email or text that to me. Okay, so that's it for video 12. Next week we're on to more cellular functions. So we're going to go into detail in some things that are happening in the cell membrane, and then I believe we get into energy systems. So how cells make ATP. So that's it for this video, and I will see you in class next week.